Hi guys, my name is Nakaima Angel. Welcome to Nsanji Media. I do BCM ICT. With me, I'm with my colleagues Mugume Mugabi Esther, Nanyange Angela, Nakandi Juliet Dabrat, Mukwaya Tanas, and Muyanja Aldrin. We are here to interest you in different topics of biology as for students offering BCM and PCB students. Under the topics, we are going to look about reproduction, we have coordination, homeostasis, and osmoregulation. Guys, I hope you enjoy the show.
Hi Nsanji Media, I'm by names of Mugume Mugabi Esther, and I'm going to discuss about homeostasis. What is homeostasis? Homeostasis, this is the maintenance of a constant internal environment at optimal constant levels. Now, all the homeostatic processes, they are controlled by a negative feedback. Yes, there is a positive feedback, but most of the homeostatic processes are controlled by a negative feedback. Okay, we have the components of a homeostatic system. We have the receptors, we have the control centers, and we have the effectors. Those are the components. So I'm going to give examples of each. Under the receptors, we have the skin and the hypothalamus, and under the control centers we have the brain, but particularly the hypothalamus and the pituitary gland. And under the effectors, we have the skin, we have the liver, we have the skeletal muscles, we have the adrenal gland, and we have the thyroid gland. You can add others if you have them. And today I'm going to discuss about the temperature regulation of a body in a cold environment. That's the main emphasis of this discussion about homeostasis. So, skin has thermoreceptors. When I talk about anything about thermal, we are talking about temperature. So, skin has thermoreceptors which detect the changes in the outside environment. And they generate, those thermoreceptors, they generate nerve impulses and send them to the hypothalamus, which stimulates or activates the heat gain center. Remember, we are in a cold environment, so we need more heat in our body to survive. Okay, now after sending to the nerve impulses to the heat gain to the hypothalamus, and the hypothalamus has activated the heat gain center, so what comes next? Now there is contraction of the erector pili muscles. They are under the skin, yeah? Those ones, and this causes the rising of the hair on the skin, trapping a layer of warm air, insulating the body from cold air. I guess you've understood that, yeah? Yes. Okay. So, and in a cold environment, people don't sweat unless you have a problem with your body, but people don't sweat. So, sweat production stops. No sweating in a cold environment. No sweating. So sweat production stops. And the skin temperature increases. Remember we are maintaining a heat in our body. We are in a cold environment. So to survive we need heat. The constriction of the blood vessels occurs. The blood vessels which I'm talking about, those under the skin. And this leads to a decrease in the diameter of these blood vessels, thus leading to the decrease in the blood flow to the skin, therefore leading to a decrease in the heat loss. Therefore, you are maintaining heat in the body. Okay, now shivering. Some people see others shivering and they say, what's that? Now we're going to look at it briefly because I'm not the one who invented how it started. Okay, shivering is due to the contraction of the skeletal muscles. Therefore, this leads to the generation of heat. You know, when you're shivering, the body generates heat, and shivering is caused by the contraction of the skeletal muscles. Remember, I said that the skeletal muscles. Yeah. Remember, I said that the skeletal muscles, they are under the effectors, and the effectors are under the components of the homeostatic system. Okay, we go on. Now, I've finished everything about the skin. Now we are going to go to other effectors. I talked about the thyroid gland and the adrenal gland. So nerve impulses from the heat gain center of the hypothalamus are sent to the adrenal gland and the thyroid gland, and this stimulates them to secrete the adrenaline and the thyroxine, which increases in the metabolic rate. Therefore, when a metabolic rate increases, there is increase in heat, because a metabolic rate is a chemical process, and that process leads to the increase in heat.
So Esther, thank you so much for talking about the other thing was disturbing me, this for the electropure muscle. Yeah? So I couldn't differentiate where it could contract and relax. So now it contracts when we are in cold environment. It contracts the electropure muscle.
It contracts,
leading
to a raise
of the hair, then creating a layer of warm air, which insulates the body from cold air. So now I get it. Thank you so much.
Yeah, I've also learned that shivering is due to the action of the skeletal muscles, because I did not know, but right now I'm able to identify that is due to the contraction of the skeletal muscles that we are able to shiver. Thank you.
Thank you, Esther, for the information. I've also learned the components of a homeostatic system: the control center, the effectors, and the receptors. Oh, wow.
Thank you, Esther, for the wonderful discussion. But I would like to inquire, or maybe add on, if you don't mind. I would like us to you help us to differentiate between negative feedback and positive feedback. And if you don't mind, you also give us the various examples in each case. Thank you.
So a negative feedback, this is the mechanism in which the change in the variables triggers a response, which leads to the reduction in the variables, returning the system back to normal. That is the negative feedback. Whereas the positive feedback, this is the mechanism in which the change in variables of the system triggers a response, which leads to the further increase in the variables and leading the system far away from the normal. Okay.
So thank you for the good explanations, and I would like to give one of the examples, particularly of the positive feedback mechanism, because it's one of the things that interests me the most. For example, one of the good examples is during nervous coordination, where depolarization leads to increase in the permeability of the axon membrane to sodium ions, causing an influx of more sodium ions into the axon. Yes, and that one will lead to generation of an action potential, as we all know, or as we shall know later as we go into the discussion for coordination. And then the other things, I don't know whether someone else would love to add on. Thank you.
So on addition to what she has said, I have another example of positive feedback. So for me, what interests me most is the contraction of the myometrium when a woman is giving birth. So I love it so much. I know that it's painful, but I really love it. Yeah.
For me, I'm giving an example of a negative feedback mechanism. Negative feedback is more experienced during temperature regulation. For example, when temperatures go beyond optimum, there are mechanisms that are set up by the body so that to return the temperatures back to normal. And we also have water balance in blood. When water in the blood exceeds the optimum levels, different mechanisms are set up in the body so as to restore the water balance back to normal. Yes. Thank you, our previous discussant. Previously, I'm Nakaima Angel. I'm going to talk about excretion and osmoregulation. Excretion, first of all, refers to the removal of waste products of metabolism from the body. And osmoregulation refers to the maintenance of a constant osmotic environment in the body of an organism. And under excretion, we have got the kidney. The kidney is regarded as the major excretory and osmoregulatory organ in the body of an organism. And in the kidney, I'm going to specialize about the human kidney. The human kidney has got various advantages, and one of them is that it is responsible for excretion. And another thing, it's responsible for osmoregulation and so many other advantages. And the other thing is that the kidneys are in a pair. Hope we all know that, yeah? And they are situated at the back of the lower abdominal cavity of an organism. And the other thing, we are going to look at the kidney nephron. And the kidney nephron is the basic unit and the structural element of the kidney. The kidney nephron has got 6 parts, yeah? And first of all, we have under the parts of the kidney, we have the renal capsule, or the so-called the Bowman's capsule. We have the proximal convoluted tubule. We have the descending loop of Henry, the ascending loop of Henry, the distal convoluted tubule, and the collecting ducts. And each of those parts here, each of those has got an advantage. For example, the renal capsule, or the widely known as the Bowman's capsule, yeah? Its advantage, or the main advantage in the kidney, on the kidney nephron, there occurs ultrafiltration. And this, the proximal convoluted tubule, the loop of Henry, the distal convoluted tubule, there occurs the selective reabsorption. I hope you all know that. Yes. From there, we also have types of nephrons. And under types of nephrons, we have got the cortical nephrons. The cortical nephrons have shorter loops of Henry, yeah? And they work in normal conditions of water ability. Are you getting it? Yes. And we also have the juxtamedullary nephrons. Those work in abnormal conditions of water availability in blood. They have longer loops of Henry. I hope you know that, yes. Isn't it? And to look at the mechanism of blood flow through the kidney nephron. First of all, blood enters the kidney by the renal artery, which branches into finer and finer arteries before entering the glomerulus. And that is called, those finer branches, they are the so that form the afferent arteriole. Now, after they enter the glomerulus, and we already saw that in the glomerulus, there occurs ultrafiltration. Now there, filtered blood, after passing through the glomerulus, blood is filtered out to form the renal fluid. Then after blood, the filtered blood leaves the glomerulus by the efferent arteriole. Isn't it? And it flows through a network of capillaries into the cortex, which surround the proximal and the distal convoluted tubules. And if you want to know that, you will open your BS and you look at the structure of the nephron. You will see that the efferent arteriole, after leaving from the glomerulus, it comes back and passes through like that. You will observe that when you look at that structure. Then after, the capillaries of the vasa recta run parallel to the loops of Henry, and the collecting ducts in the medulla. Remember, we are on the mechanism through which blood flows through the kidney nephron. I hope you are noting here. Okay, let's continue. Then from there, from the collecting ducts into the medulla, these blood capillaries return blood containing substances which are useful to the body and the general circulation. From there, blood flow through the vasa recta is much less than through the distal convoluted tubules, and as a result, that enables a water potential gradient to be maintained in the tissues of the medulla. Then from there, all in all, what you have to just know, that in the kidney nephron, there are 2 processes that take place, that is ultrafiltration and selective reabsorption. Unless guys you have some things to add on from my discussion, or you have questions?
Yes, a question. I would like to ask, how are materials in blood filtered by the Bowman's capsule to form the glomerular filtrate?
From the glomerular filtrate. First of all, what you have to first know, that the blood vessel that brings blood to the kidney nephron, the so-called afferent arteriole, it has a larger diameter than the efferent arteriole that takes away blood. Therefore, the afferent arteriole brings large volumes of blood into the glomerulus, and remember that the glomerulus are very small capillaries. Now, therefore, that creates a higher hydrostatic pressure, leading to the filtration of different materials, like the smaller materials like water molecules, glucose, amino acids, and larger molecules like proteins. Blood cells are not filtered there. They move on into the efferent arteriole, and those components, the smaller ones, the water molecules, the glucose, the amino acids, they are the so that form the glomerular filtrate. I hope I've answered you.
Major disconcerts, I would like to ask you this question. The question goes, why is it that in case of excessive bleeding, the process of urine formation falls? I request for explanation. Thank you.
In case of excessive bleeding, the formation of the urine formation stops. First of all, in excessive bleeding, a lot of blood is lost, isn't it? Therefore, blood that is going into the blood that flows to the kidney nephron, isn't it? Blood that flows there is somehow the volumes are zikendera. Zazo zikendera the jukira oyo gwo musai gwe baleeta we guli responsible during urine formation. Kati volume zazo volume yagwo we nakendera kiba kitegeeza nti byonna ebinayambako during the formation of the glomerular filtrate have been decreased. Therefore, little or no glomerular filtrate is going to be formed. Therefore, little urine or no urine will be formed. I hope I have answered your question, isn't it? Hi
guys. I would like to thank you for your continuous support. Thank you for keeping on watching this show. And my name is Nanyange Angela, Senior 6 Science, offering BCM/ICT, a student at Sanji Secondary School. Yes, as my former colleagues mentioned, I'm gonna elaborate about coordination, the factual things about coordination, of course, and I know many of us find them disturbing, but I'm going to talk about them. Before that, I would like to welcome you back from the mock saga. I know mock was total mess, not only there, but also here, of course, but I would like to encourage you to keep up the spirits, no matter the going and no matter what's happening in the mocks. Yeah, to continue with our real business, it is about coordination, and I'm going to explain coordination in animals. We are going to talk about the basics, talk about things like resting potential, action potential, depolarization, hyperpolarization, all that sort. And to begin with, I'm going to start with the resting potential. I know many of you, most of us have already found that question somewhere whereby they're asking us what is resting potential. And according to me, I think resting potential is the potential difference across the axon membrane of a neuron at rest. And during the resting potential, the inside of the axon is negatively charged with respect to the outside. I don't know whether we are getting. There's this very funny question where they're asking us to explain how an action potential is maintained, established. I don't know, that sort of question. And basically, we talk about the processes that aid in the maintenance of a resting potential. And those are two processes. We have active transport and then passive diffusion. And these two processes work antagonistically to each other. For example, if I'm to elaborate, we can say that action potential is maintained by active transport and passive diffusion due to two factors, and one of them is that there is a difference in concentration of sodium and potassium ions across the axon membrane, and also due to the selective permeability of the axon membrane to these ions. Then we can go ahead and say that the axon membrane has the sodium potassium pump, which actively pumps sodium ions into the axon and potassium ions out of the axon. Like I was saying, active transport and passive diffusion work antagonistically to each other. And before we go into detail to know how they do that, we should also note that there are two factors that maintain resting potential or aid in the maintenance of resting potential. That is the difference in concentration of sodium and potassium ions across the axon membrane, and also the selective permeability of the axon membrane to these two ions, the sodium and potassium ions. And by the way, we also have the chloride ions, but in this case we are not going to talk about them much. We are going to just focus on the sodium and potassium ions. Yeah. So if I'm to continue, we can say that the axon membrane has the sodium potassium pump, which actively pumps sodium ions into out. Sodium ions again. So we are gonna go ahead and say that the axon membrane has the sodium potassium pump. This actively pumps sodium ions out of the axon and potassium ions into the axon. This will increase the concentration of the sodium ions outside the axon and also increase the concentration of potassium ions inside the axon. This active transport, however, it is going to be opposed by, like I said, active transport and passive diffusion work antagonistically. So the working of the sodium potassium pump is going to be opposed by passive diffusion of ions. And you should know that at rest, the axon membrane is more permeable to the outward diffusion of potassium ions than the inward diffusion of sodium ions. This will cause the potassium ions to rapidly diffuse out of the axon membrane, creating a negative charge inside the axon with respect to the outside, and basically it will have maintained or gained the resting potential, because remember we mentioned that during resting potential the inside is negatively charged and the outside is positively charged. So after gaining that means that we have already reached the climax of that question in simple terms. I hope we are together. So to continue, we also have another potential, and this is known as the action potential. The action potential is basically the potential difference across the axon membrane of a neuron when it is conducting an impulse. So basically you can say that action potential is a potential difference across the axon membrane of a conducting neuron. And during generation of an action potential, the inside is always positively charged with respect to the outside. So the inside is positively charged now, because in resting potential it was negative, and now during action potential it is positively charged. So you know that difference. So we are going to go ahead to explain how an action potential is generated. And an action potential is generated, remember we are talking about coordination. So the only way, the resting potential, it is the normal state of an axon membrane when it's not doing anything. So here in action potential we are talking about an instance where we are conducting an impulse, or we are conducting an impulse, a nerve impulse. So it means that the only way we can conduct a nerve impulse, just like the former discussant mentioned, is when like a stimulus arrives. So if you talk about generation of an action potential, we shall say that upon stimulation, the sodium ion channels open and sodium ions diffuse into the axon along their concentration gradient. This will increase the concentration of the positive ions inside the axon with respect to the outside, leading to a process known as the depolarization. When depolarization occurs, it will increase the permeability of the axon membrane to sodium ions by the positive feedback mechanism. And this will now result into an influx of sodium ions into the axon, causing further depolarization until when a threshold is reached and an action potential is generated. As simple as that. Yes. I'm so sorry I never talked about this, but there are very common words in coordination, especially in this section we are looking at. There are definitions. We have what we call polarization, have what we call depolarization, we have what we call a polarized axon, a depolarized axon, we have what we call hyperpolarization, all of them. There are various definitions we find in this topic. So if you don't mind, I would like to elaborate more about them. So let me start with depolarization. So depolarization is basically the loss of a negative charge inside the axon, which is replaced by a positive charge due to excessive diffusion of sodium ions into the axon. That is depolarization. It's just basically the loss of the negative charge inside, replacing it with a positive charge. And this positive charge only comes in play when sodium ions diffuse into the axon after stimulation. And the other definition is polarization. Polarization, so it is just the opposite of depolarization. So polarization is now the gain of a negative charge, or simply the loss of a positive charge, which is replaced by the negative charge inside the axon. And this is due to the diffusion of potassium ions outside the axon. Yeah, so when they diffuse out, meaning that we are making the inside more negative with respect to the outside. So that is a depolarization. And a polarized membrane is just that, a polarized axon is an axon whose inside is negatively charged with respect to the outside, and the depolarized axon is an axon whose inside is positively charged with respect to the outside of the axon. Yes, so those are the terms. Hyperpolarization is a little bit complex. It will require us to first go through the process of regenerating the resting potential. And if you don't mind, I would like to continue with that. So, we might be asked to explain how resting potential is regenerated after an action potential has been fired. And see, after a nerve impulse has been fired, we need to bring back our axon membrane to its normal state in preparation for another stimulus. So meaning that we have to restore the original resting potential we found there before disorganizing it with the stimulus. So, and how does this occur? This occurs for us to regenerate the resting potential, we have to open the potassium ion channels to cause potassium ions to move out of the axon, and this is normally coupled with the closure of the sodium ion channels to prevent diffusion of sodium ions into the axon. So you have to also note that especially during this topic, we are going to realize that we shall only be talking about the diffusion of sodium ions into the axon, but never out. I don't know whether you are getting my concept. That sodium ions only move in by diffusion, and if they by chance are to move out of the axon, now they will be requiring force, and that's where the sodium potassium pump sets in. And similarly, the potassium ions, on the other hand, freely diffuse out of the axon, but if they are to diffuse into the axon, they need force. That is the sodium potassium pump. So for us to bring about repolarization or regeneration of the action potential, we have to open the potassium ion channels to cause potassium ions to diffuse out of the axon, and to close the sodium channels to prevent more sodium ions from entering into the axon. So remember, as we are firing our action potential, sodium ions had moved into the axon, meaning that we shall need now the sodium potassium pump to forcefully take them out. I hope you are getting my point. So when now we have our potassium ions start to move out of the axon, we are going to make the inside of the axon more negative because we are losing the positive charge inside. The negativity of the axon membrane is not as a result of bringing in any negative ion. No, it is just as a result of withdrawing or taking out the positive charge. So as we continuously take out the positive potassium ions by diffusion, the inside of the axon will now become negatively charged. However, there are circumstances where there is a delay in closure of the potassium ion channels, and this will cause excessive loss of potassium ions from the axon to the outside. And this is termed as hyperpolarization. So I get back to the other definition we had skipped up. And see, hyperpolarization, this one is just the change in the neuron's resting potential into a more negative potential due to excessive loss of potassium ions from the axon. I hope you are getting it. That when, remember, it's true we want to get a negative potential because we know that normal resting potential is about -70. But if we delay to close the potassium ion channels, we are going to exceed that -70 and we go into the -80s and so on. And that phenomenon is just known as hyperpolarization. So in order for the axon to rectify the error, it's now the sodium potassium pump will reset in to actively pump a few potassium ions back into the axon to restore the normal resting potential. And in that way, we have gone back to the normal resting potential, and the axon or the neuron is now ready to transmit another impulse upon the next stimulation. And that's it for now. Thanks for listening to me, and I would like to invite any questions, supplements, subtractions, anything. Thank you.
Thank you, Angela, for what you've discussed. And I have a question. Can you explain to us the significance of the nervous system to an organism? Because I've ever seen that question and I would like to know how I can approach it.
Thank you, Angela, for the discussion. So for me, I want you to help me and repeat for me the process of hyperpolarization. That capacity there.
For that, for me, I want you to clarify more about depolarization and repolarization.
For that discussion, I'd briefly like you to elaborate about refractory period.
I would like to begin with Angel's question. She's asking me to elaborate more on hyperpolarization, right? So, as I said before, hyperpolarization is just a change in the neuron's resting potential into a more negative potential. For example, we have a resting potential, is it of -70 millivolts? So, if we are to talk about hyperpolarization, it means that we are even exceeding the -70, going into -80, going into -90 or beyond. That is basically hyperpolarization. And this is how it occurs, I repeat. It occurs due to the delay in closure of the potassium ion channels, causing excessive loss of potassium ions from the axon. Like more of the potassium ions than what was planned is actually lost from the axon to the outside. So this makes the inside to become increasingly more negative than the resting potential. And as I say, the good news is that the system now readjusts itself again, and it sets in the sodium potassium pump to now pump back some of those lost potassium ions back into the axon to restore the original resting potential. Just like you can relate to the graph of the potentials in the axon. It's there in the box. That's it. I hope I have answered your question. Okay. So the next question, someone wanted me to clarify about repolarization and depolarization. And I'm going to start with depolarization. Depolarization is just the loss of the negative charge inside the axon being replaced by a positive charge due to the diffusion of sodium ions into the axon. Depolarization means that we are losing our polarized state, and we know that a polarized axon is one which has its inside negatively charged and the outside positively charged. So meaning that if we are depolarizing, it means that we are changing the state into its vice versa, that now we are making the inside to be positive and the outside to be negative. I think that is it. And repolarization, what I can talk about it is that repolarization is a way of bringing back or re-establishing the resting potential. This is in that initially we had our resting potential, and when the stimulus arrived, we had generation of an action potential and so on. So meaning that the membrane has lost its polarized state. So in repolarization, we are trying to bring back the resting potential. Because now in this case, after transmitting our impulse, the inside is still positively charged and the outside is negatively charged. So in repolarization, we are remaking the inside negatively charged and remaking the outside positively charged. That's all I can say about repolarization. I hope we are together. And someone here asked me to clarify about refractory period. So refractory period is basically the period of inexcitability of an axon membrane. Like it is not able to transmit an impulse. That is refractory period. It is in its resting phase. And the refractory period is in two categories. We have the absolute refractory period and the relative refractory period. And during the absolute refractory period, no matter the intensity of the stimulus, an action potential cannot be generated. It does not matter how intense the arriving stimulus is, because the reason is because during absolute refractory period, almost all of the sodium ions are inside the axon, meaning that even though a stimulus arrives, yes, it will result into opening of the sodium gated channels, but there will be no sodium ions to move into the axon because they are all already inside. I hope we are getting. So during in that phase, we shall not generate an action potential no matter how intense our stimulus is. Then the other one is relative refractory period. So now during relative refractory period, if the arriving stimulus is of greater intensity than the normal, we shall generate an action potential. Because during this phase, some of the sodium ions have been pumped back by the sodium potassium pump outside the axon, meaning that when the stimulus arrives and it is of much more intensity, and it will cause the opening of some of the sodium ion channels, and those few sodium ions will diffuse into the axon to generate an action potential. So basically that's what I can talk about the refractory period. Thank you. The last question, someone was asking me to elaborate about the significance of the nervous system in coordination, right? And what I know is that we are only going to talk about the basic components of the nervous system and how they work together to bring about coordination. The components include the receptors, you get. Then we have the neurons, that is the sensory and the motor neurons. Basically, although the relay neurons can also be parts. We also have the central nervous system and we have the effectors. So if we are to talk about the significance of the nervous system in coordination, we are just going to bring together all those components and what they do in order to bring about coordination. I hope we are together.